Give me just 10 minutes and I'll explain to you the entire glycolysis process. So if that's something that interests you, let's start. So what is actually glycolysis? Glycolysis is simply a biochemical reaction that occurs within the cytoplasm of cells where glucose, a 6-carbon containing molecule, is being broken down to 2 molecules, each containing 3 carbons. And this molecule is called pyruvate. So that is the end product of glycolysis. Now, the way I actually like to see glycolysis is like glycolysis is a process, is like an engine saw that is in charge of breaking down a log of wood, which is in this case glucose, into smaller pieces called pyruvate. Now, the reason why this is important is because if you use glucose just like that, you are going to use more energy just to get it to produce energy, and that's not efficient. And that is why glycolysis is here to break it down to pyruvate, which is far more usable in the next respiratory process. So that's one reason and importance of glycolysis. Now, glycolysis is a 10-step biochemical pathway. So I've divided these phases, these pathways into 2 phases: the energy investing phase and the PL phase. In the energy investing phase, we use some ATP that is from the body, and in PL phase we produce more ATP, right, into the body and a pyruvate. So let's begin the first reaction. The first reaction is a reaction where we are trapping glucose. We are trying to trap glucose within the cell. Now, the enzyme that does this is known as hexokinase, where it takes a phosphate molecule from ATP, which is the cofactor, and adds it onto glucose to form glucose 6-phosphate. Now, glucose cannot leave the cell. Glucose cannot leave the cell, preparing it for the next phosphorylation phase. Now, we have to convert our glucose 6-phosphate into fructose 6-phosphate, which is far more reactive and unstable. So the enzyme that does that is phosphoglucose isomerase. Why? Because this glucose 6-phosphate is a phosphoglucose. So we are converting our aldose phosphate to a ketose phosphate. Now this product is far more reactive than glucose 6-phosphate. So this makes the next process far more easier. Now, the last reaction in the energy investing phase is where we produce fructose 1,6-biphosphate. Now, this step is also the rate limiting step of glycolysis, which means after this step, glycolysis cannot be stopped. And it's within this step that we can regulate glycolysis, maybe through the amount of ATP that is already made or by hormonal action. So the enzyme that does that is phosphofructokinase 1. Why? Because it adds the phosphate onto the first carbon on fructose. Now, don't forget the cofactor, which is ATP. So in the energy payoff phase, the first reaction here, we have now to split our highly rich molecule we made in our energy investing phase into 2 molecules called, the first one called dihydroacetone phosphate, and the second one called glyceraldehyde 3-phosphate. Now, this is far more simple because fructose 1,6-biphosphate is energy rich and very unstable. So it can easily be broken down by the enzyme aldolase. Now, glucose prefers triglyceraldehyde 3-phosphate for the subsequent reactions. So we have to convert that dihydroacetone to glyceraldehyde 3-phosphate. Now, thankfully, dihydroacetone phosphate is an isomer to glyceraldehyde 3-phosphate. So the enzyme that isomerizes it to G3P is known as a triose phosphate because this is a triose. It has 3 carbons, remember. So this is a triophosphate isomerase. So now anything that occurs after reaction 5 occurs twice. So in reaction 6, glyceraldehyde 3-phosphate is being oxidized, where we remove a hydrogen atom from it and add it to NAD to form NADH. Now, many people might ask, then where does the extra phosphate come from? It doesn't come from ATP, right? That is an organic phosphate, so it comes from an inorganic source. So that is why you have the product to be 1,3-biphosphoglycerate. Now, this is the first reaction that leads to an energy formed. So note that that's very, very key. So the next reaction is our first substrate level phosphorylation, where we make ATP from a substrate. Okay, we make ATP directly from a substrate. So the enzyme that removes a phosphate from 1,3-biphosphogylucerate to form 3-phosphoglycerate is known as phosphoglycerate kinase. You should just see kinase as those enzymes in charge of removing phosphate in the case of glycolysis. So any enzyme that removes phosphate, when you hear phosphate has been removed, think of a kinase. Then just think of the product. What is the product? The product is a phosphoglycerate. So that is why you have in this reaction 3 phosphoglycerate being made. Now remember, it occurs twice. So which means in reaction 6 we have 2 NADH produced, and in reaction 7 we have 2 ATP produced, meaning that we are now balanced, right? So in reaction 8 we have to rearrange the phosphate that is on the 3-phosphoglycerate. So the way I like to think about this enzyme, the enzyme that does this, like, all right, if you have a mutation in a gene, what does that mean? That is a mutation. So the enzyme here is a mutase. So what is the substrate? It's a phosphoglycerate. So it's a phosphoglycerate mutase where 3-phosphoglycerate is being converted to 2-phosphoglycerate. Now reaction 9, we will form an enol from 2-phosphoglycerate. How does this occur? We just simply have to remove water from 2-phosphoglycerate. So the enzyme that does this is known as enolase. So when you remove water from 2-phosphoglycerate, you have phosphoenol pyruvate. Now phosphoenol pyruvate is very energy rich. It's very, very rich in energy, and it can easily donate its phosphate to form pyruvate. And that is why the last reaction in glycolysis is where we form pyruvate from PET, that is phosphoenol pyruvate. So we just simply have to remove the phosphate that is on PET to form pyruvate. And once you do that, you have pyruvate, and the enzyme that does that is a kinase. And what is this substrate? Enol pyruvate. So you have pyruvate kinase. So this is a second substrate level phosphorylation because you make ATP directly. So in this reaction, 2 ATP is gained. So this means directly from glycolysis you make 2 ATP. But if you include the 2 NADH we made after they pass through the electron transport system, this is simply mean glycolysis gives us a net ATP of 8 because 1 NADH leads to the production of 3 ATP. Now, some important information you have to know is the yield from glycolysis, which I've already explained. So end product is 2 pyruvate molecules, 2 ATP made directly, and 2 NADH that is made. Now, why is it important for us to know glycolysis? It's important for us to master glycolysis because some cells do not have mitochondria, such as blood cells. This means that glycolysis is the main way where these cells produce energy that they use for their metabolism. And only that, tumors, cancer cells, grow very, very fast. They grow very, very fast, and because of that, they do not necessarily need mitochondria. So that is a very key research nowadays where we are trying to use this principle to kill cancer cells. Maybe it's already been used. I'm pretty sure. I haven't actually read the papers on that yet. And only that, in anaerobic condition where oxygen is small in circumstances where we are doing intensive exercise, what we realize is that the pyruvate form will be broken down to lactate, which will be further broken down to the pyruvate will be broken down to lactic acid, which will be further being broken down to lactate and a proton. Now this will lead to an acidosis because we are pumping in more protons within the body. So it's also very, very key because sometimes patients used to have acidosis. So you get why it's important for you to learn glycolysis. Well, that is that for glycolysis under 10 minutes. If you have derived any sort of value from this video, you might want to like or share to a friend who is somehow struggling to understand the whole process of glycolysis. Until then, have a nice day, stay blessed guys, and see you in the next one.